Hi everyone, I am Saniya. This is cute little Yangshi, and this is cute little me. And this video, we'll be talking about how we can use gProfiler to translate gene identifiers between various organisms. Like the little badger here, for instance. Today, I want to talk to you about our DNA basically, and how we have different genes, but how there's some similarities between like our human genes and these cute little cats and their genes. The genetic diversity, there are a lot of overlaps between us. And as you'll find out, the cats from my example this time, actually it was so random. I just selected 59 random genes with their IDs, and I tried mapping them to genes in various organisms, beginning with cats, then their favorite treat, which are mice, and lastly the cows. And we had a winner, and the winner was cats. Cats had the most number of genes that mapped between our list of human genes and the list of cat genes as well, out of the 59 that I input. So it's really cool because we can figure out from different experiments how different genes in one organism map to genes in others. Do they have the same name? Do they have different name? Do they not map at all? And this makes science more translatable. There are various tools out there. I just wanted to show you a very intuitive one that I discovered that uses gProfiler that I've already kind of talked about in other videos as well. So I hope that this will be a helpful tool for you guys to be able to convert between different like species in terms of their gene names, figuring out what works and how genes in my DNA, me and Yangshi, how those are actually like related to each other. Because I'm definitely not as cute as these cute little cats here. So if you ever have a list of genes, with a numeric ID form, for instance, like entree IDs or ensemble IDs, like how do you figure out what gene names and IDs those map to in other organisms? That's what we'll be talking about here. Like for instance, what if I want to convert between my list of gene names and figure out in cute little me what those correspond to? How would I do that? So that's what we are going to do. So we're going to have a numeric list of gene names. And that's why I sort of stress having the entree gene ID or other identifications, because some genes names, like they could map to the same entree ID potentially, because people use different aliases for a gene. And I have other videos talking about those as well. But you want to be able to map a list of gene IDs from one organism to the corresponding ones in another organism. So you can figure that out for little cute little May there. One really cool data resource that I learned about is it's called Mango, which is the mammalian adult neurogenesis gene ontology. So it's not a mango like this. It's not really that ripe anyways. But when I talk about mango, I'm talking about the gene ontology for neurogenesis genes, especially genes that have been recently found, especially in regions like the hippocampus of the brain, to be involved in creating new neurons. So what you'll see from this really cool data resource here is that it's a mammalian adult neurogenesis gene ontology, and it has a bunch of different species like guinea pig, human, monkey, mouse, rat, and other. And these experiments done in vivo in living organisms, especially for the rat, mouse. If we look at the human as well, so these are different studies. And these are like around 1,670 entries. These are different types, like neurons that are new neurons, precursors, immature neurons, all of these types of things. And it's a really cool database and resource as well. They try to explain these things as well. What are the genes that are used? What is the ontology? This is the ontology. So an adult neurogenic cell, it's able to create new neurons in the brain. It's precursors, progenitors, late progenitors, immature neurons, mature neurons, astrocytes, and so on. But what you're going to notice here is that it has different gene names that correspond to different species like mouse and rat and pig and human. We have different databases. In fact, there is a little database of names for mice. There is the MGI, it's a mouse nomenclature homepage. Oh, luckily May won't be seeing those mouse photos. Otherwise, those would be pretty activating for me. But what you're going to notice is that we're going to be talking basically about how we can do this. So in short, if we have gene entry IDs, which or any sort of identifier can be used here, how can we map from one organism to another? Like for instance, how can we map from human to the Felis catus, which is a cat apparently. These are the three little cats. This is Yangshi, that's me, and this is Toggle. And like this is a phylogenetic tree, and an animal phylogeny, for instance, as well. And how can you translate like these human identifiers here from one species to the next? So if we were looking at this Mango database and see from like these humans to the cats here, like in Boss Baby, it's a very cute show. So we're going to be talking basically about gProfiler, and I have other videos talking about functional profiling, SNP ID to gene name. I have other uses for here. Here, there's unfortunately no example query here that as of now, but we can still talk about how we would go about this. So we have different nomenclatures for genes. We have genes here. This is the mouse database as well. We can hunt through different genes. So for instance, like this ABCA2, it's involved in expression in a mouse. And so many times we do these studies in mice or other organisms because they're so, many times we do these studies in mice or other organisms because they're so involved, and they involve a lot of risks, and they might take generations for us to see their effects. So, usually these species, their generations are much shorter, and we're able to do experiments for them. So now we're going to navigate here basically to gProfiler. But what I want to show you first, I want to show you why it is what it is here. So we're going to gOrth, which is orthology search, this one right here. So it translates gene identifiers between organisms. We provide orthologous gene mappings based on the information we retrieve from the Ensemble database. So it's like you can think of it again, kind of like a phylogenetic tree of species. You can think of it that way, where how these species are mapping, and it's trying to map and figure out the gene names, gene IDs as well. So humans here, and then you can do mouse mapping. So we can do so many mappings. So since we have a cute little cat sleeping on the back right here, we can just do cat. Felis catus, for instance. And we can put in wiki genes, KEGG enzymes, Illumina, BioGrid, Affymetrix. I think these are like microarrays. So we're going to do for Entrez gene. So what an Entrez gene is, so let's look up this APO gene. If we look up a gene, for instance, the genes have different identifiers, HGNC symbol, NCBI, Entrez ID, which is what I'm a huge fan of, the Ensemble ID, OMIM, which I think is the is for mice, and UniPro, KB, Swiss Prot. We have all of this over here, but for this gene APO, we're going to be using N348 for the Entrez ID, for instance. So similarly, I have this file of just some random gene names that I wanted to run through. So in this list, we have around 59 different gene names and the corresponding Entrez IDs. So for instance, if we looked up this gene here, SNHG24, we should expect that its Entrez ID is 79104, just to kind of show you guys again, like what this means exactly. And of course, you see that it mapped to something else. So what I had put in is I had put in SNHG24, which is what this is here. It's a previous name, and it got updated to MEG8. So guys, this should really motivate why we don't want to always stick with gene names, because those are outdated. And I spent a lot of time creating a mapping file that I've talked about in other videos, how you can use that to go from gene names to entry IDs. But this just shows you how you might have a very outdated gene name and they get updated sometimes based on functions and other things that then maybe they get withdrawn from databases. So you want to be careful here. And now you see here how I've mapped essentially like this to this gene ID here, which corresponds actually to MEG8. So this 79104 corresponds to MEG8, but it is acknowledged that it's a previous, it is a previous name for this gene. So for instance, what you're going to see is that this is the SNHG24, which is what this is over here. And it corresponds to the ID that I've identified here, which is 79104. Okay. Which is what this is here, and it's one updated gene. So anyways, these are all the ones, are all the respective IDs that I looked up and I mapped them to. And again, there are other videos that I've gone through where, again, you can see what the mappings would be. Like NO7P1, let's see what this is. And you can see again here, this is another example of this gene being updated as well. So it used to be called NO7P1, which is what we had. And what we can see here is that NO7P1 actually maps to 101927546, which is what this is. So this maps to this, which is great. And again, this NO7P1 was a previous name for this gene here, the previous name. And the new name is NO7L1. So this again is another example that kind of shows you how, but so usually we hope that it's the same one, that it will fall under this name here, but sometimes that's not the case. So, you know, because the gene has been updated in terms of its name. So again, you'll see, you just need to find it somewhere on this, on top half of this page where it talks about, you just really need to ensure that the gene of interest is somewhere like in this part here. That it's somewhere in this part. It's either directly mentioned in aliases for the gene or previous names. You just need to make sure it's here. So, okay, so that being said, I'm going to show you why we want to go through the IDs, especially because if we're using this tool, we kind of have to stick to what the requirements are of the tool that we're using. And many times I notice that tools tend to use entry IDs, or those can be more robust. So for this list of 59 genes, let's just go here in gProfiler, and let's just say that, like, let's use the gene names here. Let's just copy these gene names. And these are for humans. So let me just also mention that here. These are all like for human entry IDs. These are all humans. So let's go down and copy these. And then let's see what happens if we try to map them to the cat, for instance. But and now we see numeric IDs treated as, there's no numeric ID here. So if we run the query, we're going to get nothing because it's not numeric at all. It needs numeric IDs. So what we're going to do is we're going to go back here, and I'm going to just take the respective entry IDs instead. And I'm going to rerun this analysis. I'm going to wipe everything out here. I'm just going to delete. I'm just going to delete this and run this query here. But before I do that, actually, numeric ID is treated as Entres gene extension. That's what it is. I'm using Entres gene. That's what we were using here. We were using the Entres gene. You can use other ones too. You can use if you have like KEGG, Wikigene, these other ones, you can use this, but Entres gene is pretty reliable, I would say. You know, I mean, it's much simpler. I feel like I'm not really familiar with the others as much. So we're going to run the query, and now you see that we get results here for many of them. But if there are some that don't map to any cat genes, then they're just NA. There's no ortholog if they can't find it. Or they try to fill it up as much as possible. And we can export this to a CSV that we can use and analyze, but we can see for certain genes. Like for instance, this gene right here, this 100170841 that is like C17 Orth, origin replication factor 96. This maps to, in a cat, it is mapped to the EPOP gene. So whereas this is in humans, in cats, it maps to EPOP in cats, for instance. And then this gene 811554, which is WBSCR16 in humans, maps to, so it maps to instead RCC1L in cats. So this is important because when we do experiments like in brain diseases or other experiments with mice, for instance, or other creatures, let me just show you, these poor guys get used a lot. When we run these experiments with these lab mice, here, when we run them with these poor little mice here, May still sleeping. Otherwise, May would have been very activated by this. But when we run these experiments, these poor guys, right? Like, so we will get certain genes, maybe, or biomarker genes that are key for mice, for instance. And we may want to map them back to what those could be in humans and maybe see if those are risk genes in humans, for clinical trial development and whatnot. But you can all see like how we've gone from this ID, gene ID, which again, like 757223 is the next one, which is SMEK2, and it maps to PPP4 4R3B. So again, we're just sort of like doing some intersection between this and these over here. And just to make this a little bit more robust, actually, I'll just show you how we can do this with, some VLOOKUPs as well, how you can just, take this stuff back that we've gotten and apply it. So we have this here, we're going to open this up. So it's really cool because it tells you exactly the time and date and what organism. So Homo sapiens, H sapiens is the source, which is, where our initial gene IDs were from humans, Homo sapiens. Homo sapiens is humans. Human, Homo sapien. That was what we originally had. And our source is this, cat, feline cat, this or so. So that's what we had before. This is our source. And this is the mapping that we've gotten from human to cat for this list of, initial aliases. So what we could do now is we can just take this. For instance, we can move our original list here, and we can take this list that we've gotten. We can ignore what this is for now, but we can just take this list. And, all of these map to this, so like, equals. Yep, true. There are some that were not found here. So some of them could have like multiple ones here that you'd have to be cognizant of. So what we could do is we can just say, okay, this is the initial, so this is initial alias. So this is the human Entrez ID. And we can just put the gene name because this may not be intuitive for you. So this is the human gene name. And what we also have to do here is we just have to do a VLOOKUP here where we're just looking up, so we can arrange it this way. But what we're going to do is we're just going to look up here. We're just going to look up. So it's called a VLOOKUP. So we have the human entry IDs, and because some of them have multiple rows, it's not quite the same mapping per se. So all we're going to do is we're just going to VLOOKUP this entry ID in this set of data here. So it's important that whatever you're looking up, it has to be the leftmost column in whatever you're selecting. We're selecting columns A to B, and we're saying, please look up at element H2 in the set of columns A, B, and then please return the second column, so this is column 1, and this is column 2. So we're saying, please return the second column, and you can do false if you want an exact match. So this is how I've done. I'm looking up this here. I'm looking it up in this here, and I'm doing false. So I got better at this with a lot of practice. I used to dread VLOOKUPs. So now you can sort of see that, and then we can just copy this and then do a paste special, because what we had is these were formulas, and we just want to transfer them here. So now what you can see is that we have the human entry ID and the human gene name. And this is, it gives you the initial ensemble ID as well for those genes that it found. And it gives you the ortholog name. So this is the cat gene name. And that's what this is. And it'll also try to tell you the explanation if it is able to find them. So we've now gone from the human gene name to the cat gene name, which is pretty cool. So now I can see from me, who's fast asleep, how we can go from the human gene name all the way to the cat gene name. So basically, what I'm trying to show Yankee and me, the cute little siblings, is how I can go from a list of my gene names. You know, how I can go from a list of my gene names, Yankee and me, to figuring out what the genes are called for you guys. You guys are cute little cats, right? So that's pretty cool, right? Wouldn't you say that? So that's what we're able to do. We're able to do that mapping. We're able to see, you know, for a list of gene names, you know, in my human DNA, what those are for you guys. Do you guys have the same genes or not? And if so, what are those called for you guys? That's pretty cool. So that's what we found here, Yankee. So what we're finding here, as I was showing you guys, is that, you know, we can go from a human gene name to the cat gene name here if we're able to find them. So like, you know, SNHG24 is unknown, but for instance, this one is some GPR 110 maps to EADRF1, which is some adhesion G protein coupled receptor F1. So you have all these proteins and genes, Yankee. You're very complex. And me, you guys are very, very complex creatures. Very complex. This maps to this. And for instance, EMR1 maps to ADREG1 as well. So there's so many mappings that we can do. Up next, what we're going to see is how we can map not just to cats, but humans to mice. I'm going to show you some really cool images of mice right now. And let's see if you get activated. You see these images of mice? You see? Oh, he really wants to eat these little mice or play with them. See, look at these cute little mice. What genes do I have in common with those, huh? So that's what I want to do here. I just want to show you guys another example where I'm basically going to be looking at the gene names and instead of looking at them for mice. So again, what this just shows you is how we go from human gene names, but especially using entry ID or some numeric identifier to the cat names, gene names that are mapped to it. So we just get rid of all like the NAs, and then we can just find the ones that are mapped. We just get rid of all the not applicable. And then we have this list of, out of those 59 ones that we had, there are some, okay, so we have some repeats here. So we can see here that this gene maps to many of them. So I just want to show you, is out of the 59, let's just see which ones map properly here. So just so that we can see how many did I have in common with you guys. That's what we want to see. So I'm going to remove these and like, let me just remove the duplicates and let's see how many I had in common. So I had a list of 59 genes from before. I had this list of 59 unique genes here, 59 unique genes. And from this list of 59 genes, only 26 are found in you, Yangshi. Yangshi, you only have 26 of those genes. What happened to the other, huh? There are another 33 genes that did not map, but we can see the ones that did. And those are the ortholog names for us to use. So again, this was for humans to cats, but what about for human to mice? Right, we should see what we can find for human to mice, for instance. So I'm going to try to rerun this analysis. And instead, what we're going to try to do is we're going to focus instead on mouse. Mus musculus, mouse. We're going to run this query and we're going to see that there's some differences. Well, EPOP is the same, RCCL, TRAP 10, C10, okay. So they're a nectin 4. So there's some differences here. What we're going to see is that, you know, there's nectin 4, TRAP C10. So this is sort of like what we get when we're running this analysis for like mus musculus, for instance. So we take these IDs and we can map them to mus musculus, to this organism for mice, see what we find here, see if there's more overlap or not. So let's export this to CSV and then see how many of the aliases we found. So if we download this for the mice, what we're going to get rid of is we're going to get rid of all the ones here that were not mapped. So let's see how many of them mapped. So what we can do is just get rid of all of those that had NA here that did not map in the end. Those that had NA, we're just going to remove those. And let's see how many of those mapped. And we might want to see as well, these are the aliases. There might be some overlap here. So it seems like there was a bit fewer overlap here for the humans and mice here, the 24. So that means that there was a little bit more similarity here. So the 23 of them mapped between humans and mice instead. And I think that there were 26 that mapped between the human and the cat. So 26 for human and cat for this list of genes here. And there were just 23 that map between the human and mouse. So thus, Yankee, there is more overlap between you and me than between me and a mouse. That's pretty cool, right? I hope that this was really helpful for you guys to see how we can go from a list of gene names for like humans or any species in general and map between each other, see which genes align with each other, which genes are different, and if different, then, you know, which ones are found in which organism with the same name, which ones are found in with a different name in a different organism. So through this analysis, we can take a list of gene IDs or some sort of way, you know, from gene names, we can extract the list of IDs like entree IDs. I'm a huge fan. And we can go from there to mapping, like whether it's human entree IDs or something else. We can go from that to ultimately mapping to some other organism like cats here, or like mice or cows or so many other species. We can go and do that kind of analysis, mapping between our list of gene IDs, for instance, and another organism, and seeing what they have in common, which genes have the same name in the different species, which have different names, which ones don't even map at all. But it gives you a way, because when we get findings from different experiments for organisms, we'd like to see which ones are interchangeable, and I just will show you, like, what if we want to, lastly, try to figure out what are like overlaps between humans and not just mice, and maybe like Bos taurus, which is a cow. What if we want to figure this out for the cow? After all, like I was showing a little cow here. What if we want to do that analysis? So yeah, we're gonna see that, this is what happens when we're mapping it to the cow. It seems like, the Selenov, KC and E5, some of the different as well. So if we export this to the CSV, we can see, so this is human to mouse, this is human to cow. Then we can just focus on the aliases that are mapped. So we can get rid of those that are NA. We can just remove those that are NA. And then just remove some duplicates in case some of the on-readies happened. So the 26 actually. So we have like 26 genes. Actually, this is 25. So 25 that map between human and the cow here on this list. We have about 26 that map between the cat and the human and the cat on the same list. And we had, for the mouse musculus, we had like 23. So I think it's safe to say that cats are the winner. So I hope that this is helpful for you guys. So all you just need to do is take an input list of genes from any organism. Please make sure you have some sort of identifier for them, and then just have to run them through. So I'm Saniya Kuller, and if you found this helpful, please like and subscribe, and please let me know if you have any questions at all.